Okay, so welcome. Here we will be discussing on the molecular organization of enzymes. Okay, based on the complexity of the molecular organization, we can identify two kinds of enzymes. Simple enzymes, you can see it is made up of proteins alone, simple proteins alone. While complex enzymes, they are otherwise referred as conjugated enzymes or conjugated proteins. They are made up of conjugated proteins. Okay, conjugated in the sense they have a protein part plus a non-protein component, isn't it? So similarly, complex enzymes do have a protein part and a non-protein part. The structure, the organization of complex enzymes, we will be looking in detail in the coming slide. Okay, so simple enzyme is made up of only simple proteins. They are simple proteins. Fine. Now we can find two kinds of enzymes based upon again the complexity. You can see here monomeric enzyme and oligomeric enzymes. Okay, monomeric enzymes are made up of a single polypeptide. Okay, it's very small in size, molecular weight is very low, and they include ribonuclease and trypsin. Okay, sorry. Now oligomeric enzymes, on the other hand, they are made up of more than one polypeptide. Okay, each polypeptide chain, it is known as a subunit. Okay, so they are made up of more than one polypeptide chain, and hence it is known as oligomeric enzymes. Okay, the examples include lactate dehydrogenase, which we have already mentioned earlier with respect to the classification, isn't it? One of the examples. Then we have is the aspartate transcarbamylase. Okay, so these are the two enzymes examples for oligomeric enzymes. Right. Now we'll look into the detailed structure of the complex enzyme. Complex enzyme is otherwise referred as the holoenzyme, and holoenzyme is composed of apoenzyme and cofactor. We have already mentioned that complex enzymes are conjugated proteins, and we already know that conjugated proteins means those proteins which have a protein part plus a non-protein component, isn't it? Likewise, holoenzyme is formed of a protein part plus a non-protein part. Protein part is referred as apoenzyme. Protein part of the holoenzyme is referred as apoenzyme, and the non-protein part is referred as a cofactor. Okay, this cofactor, it could be inorganic or organic in nature. When it is inorganic, it could be in ionic form like sodium, potassium, magnesium, manganese, iron, zinc, cobalt, etc. Only in the presence of the what you call the ions, certain enzymes would work. Okay, those enzymes are considered to be a hollow enzyme, right? Non-protein part, the organic molecules, the non-protein organic molecules, they are referred as coenzymes. Okay, so they include what you call as coenzyme A, coenzyme Q, NAD, FAD, etc. Okay, so we can see that holoenzyme, it is made up of a protein part which is referred as a apoenzyme, a non-protein part which is referred as a cofactor. Okay, cofactor are the presence of cofactor is very important because in the case of a holoenzyme, both apoenzyme and cofactor has to be present to bring about the catalysis process. Okay, so it is very indispensable. The presence of cofactors indispensable for the activity or the action to be brought about. Okay, so if the apoenzyme and cofactor is separated, it is independently action cannot be possible. That is, independently they cannot act as an enzyme. Only when they are brought together, we have a enzyme acting. Okay, now these two, the cofactors and the apoenzyme, they bind together. It could be loose binding or tight binding, and they form a complex, what you call as a holoenzyme. Okay, when a cofactor is tightly bound to an apoenzyme, it is referred as a prosthetic group. Now we can see what the cofactors. Cofactors, as we have already mentioned, they could be either inorganic or organic. Okay, inorganic, it could be any of the ionic forms. Organic, it is non-protein organic forms, and organic cofactors, they are commonly referred as coenzymes, isn't it? Now, coenzymes, there are plenty of examples: coenzyme A, coenzyme Q, nicotinamide adenine dinucleotide, flavin adenine dinucleotide, FAD. Okay, all these are examples for coenzymes. Okay, coenzymes in combination with the apoenzyme, they act as carriers or donors of atoms or chemical groups. For example, NAD can act as an acceptor. For example, when we saw lactate dehydrogenase is a trivial name. Okay, the actual name, systematic name is lactate NAD. You can see oxidoreductase. So, lactate dehydrogenase is an enzyme which catalyzes a redox reaction wherein the substrates are lactate or the lactic acid and NAD molecule. Okay, wherein NAD accepts a hydrogen from the lactic acid and it gets converted into NADH. It reduces to NADH, right? And lactic acid, it becomes oxidized. So this is what happens, right? So we can see here, coming back, so NAD, what you call the coenzyme, it can act as carriers, acceptors or donors of certain atoms or chemical groups. I hope it is clear. NAD is one example. NAD can accept hydrogen and become reduced to form NADH. Similarly, FAD can reduce to FADH2. Okay, it can be reverse also. That is, NADH can give away hydrogen atom. It can become reduced. Okay, and it can reduce some other what you call substrate molecule. So, in the case of what you call coenzymes as a part of the holoenzyme, the coenzyme can act as acceptors, carriers, donors of atoms, for example, hydrogen, or it could be chemical group. I hope it is clear. Okay. Now, major acceptors, carriers, and donors of hydrogen atoms include NAD, FAD, and coenzyme Q. Okay, coenzyme Q on acceptance of hydrogen, it becomes CoQH2. NAD on hydrogen acceptance, it becomes NADH. FAD becomes FADH2. Okay, so it could accept hydrogen atoms. Similarly, it could also donate. Okay, FADH2 can donate hydrogen atoms and be, revert back to FAD form. NADH can give hydrogen atoms or donate hydrogen atom and become NAD. Similarly, coenzyme Q. So they are all acceptors, carriers, and donors of hydrogen atom. Now, what about coenzyme A? Coenzyme A, it acts as transferring molecule. Okay, it helps in transferring of acetyl group. Okay, acetyl group, it is CH3CO, acetyl group. So these are few examples of coenzymes which can accept, carry or donate, accept or donate, and or carry certain atom or chemical groups. I hope it is clear. Coenzymes, fine. Now, there are some coenzymes that are tightly bound to apoenzymes. Okay, those coenzymes are referred as the prosthetic group. So, cofactors, specifically the coenzymes, once it is tightly bound to the apoenzyme, those coenzymes are referred as a prosthetic group. Clear? For example, iron porphyrin complex of cytochromes and hemoglobin, the flavin part of flavoprotein. These are examples for prosthetic group. We cannot separate it. Okay, for when we are speaking about lactate dehydrogenase, lactate and NAD, they come together. Okay, for the reaction to be possible. Lactate dehydrogenase is an enzyme which require NAD to what you call oxidize the lactic acid. Okay, so during that process, lactate dehydrogenase works well when NAD is present, right? But NAD is not tightly bound to lactate dehydrogenase. Only during that enzymatic action they come together, right? So it is not tightly bound, right? So they can exist independently as well, even though it is not when independent, it cannot bring about the particular enzymatic reaction, but they do have independent existence. But prosthetic group is not like that. Okay, the prosthetic group, it is the coenzyme part which is tightly bound to the apoenzyme, and the example is that of the iron porphyrin complex found in hemoglobin. Okay, so those are prosthetic group, right? So it always remain together. Clear? Now, coenzymes are of two types or two major groups. One that participate in redox reaction, as already mentioned, that is they can accept or donate hydrogen atoms. Okay, it could be NAD. When it accepts hydrogen atom, it becomes NADH, NADP, NADPH, FAD, FADH2, FMN, FMN. Likewise, so these molecules, they actually can accept or donate hydrogen atoms and bring about redox reactions. Okay, so coenzymes participating in redox reactions, the examples are these. Clear? Now, the coenzymes that participate in transfer of chemical groups. They help in transferring chemical groups from one substrate to another, and includes coenzyme A, then thymine pyrophosphate, biotin, etc. Okay, so as already mentioned, coenzyme A, for example, it helps in transferring acetyl groups, isn't it? So you'll be learning especially the beta oxidation of fatty acids and all. You will be learning the role of this one. Okay, it helps in transferring acetyl group. So, this is about the uh, enzymes. Okay, now this is example. This is a classification. So enzyme can be divided into especially the complex enzymes. Okay, the holoenzymes and simple enzymes. Okay, simple enzymes there is only protein that is the chemical composition, while the holoenzyme it is formed of protein part and non-protein part. The protein part is referred as the apoenzyme and the non-protein part is referred as a cofactor. The cofactor it could be like prosthetic group coenzyme. Okay, it is actually inorganic and organic. The coenzyme, when it is tightly bound to apoenzyme, we call it as a prosthetic group. So this is about the classification of enzymes. I hope, sorry, what you call the molecular organization of the enzymes. I hope it is clear. Okay.